Treg cells, formerly known as T suppressor cells, have an important role in guiding the maturation of T lymphocytes and thus future immune responses. For example, in asthma, Treg cells have a role in helping T cells mature and deciding whether they will emphasize Th1 responses in the future or the Th2 responses, which will worsen asthma. In the thymus, in corpuscles known as thymic corpuscles or Hassel's corpuscles, they can help promote tolerance of self, in that if there are cytotoxic T cells which would react against self peptides, such as those produced by the beta cells in the pancreas, or there are T helper cells which would react against self peptides presented to them by macrophages and thus worsen autoimmune reactions against self, such as the beta cells in the pancreas. The Treg cells then inactivate or destroy these self-reactive T lymphocytes and thus promotes tolerance so that one's immune system would tolerate one's own self and not react against self. An important gene for these Treg cells, in fact, one of the genes which, when expressed, helps define this population, is the forkhead P3 gene. The forkhead genes are transcription factors. They produce proteins which bind to DNA and turn on genes. When this forkhead gene, the FOXP3 gene, is mutated, it causes a genetic disorder known as IPEX, also known as XLAD, in which there are a number of autoimmune and allergic conditions which result, including type 1 diabetes. Since the FOXP3 gene is located on the X chromosome, most affected individuals are male, and this disorder can result in infant or childhood mortality unless it is treated with immunosuppression and bone marrow transplants. The mutation in this essential gene for Treg cells causes a dysregulation of the immune system so that cytotoxic T cells attack self, that T helper cells promote reactions against self, and this results in congenital type 1 diabetes where the islets of Langerhans can be absent at birth.